Hi guys, this is just a quick review of genetics. So you should remember some of the basic vocabulary: genotype, or your two alleles for your trait, your genetic makeup; phenotype, which is the trait that you have because of your genotype. So your genotype determines your phenotype. Homozygous, meaning two of the same. You can be homozygous dominant or homozygous recessive. Heterozygous, having two different alleles, big T, big G, little g. So you have the dominant and you have the recessive. And an important fact is that when you have a heterozygous genotype and you're following the law of dominance, that individual will always have the dominant trait. So in this case, tall is dominant because that's the trait that you get with big T, little t. In this example, a heterozygous frog is brown. The dominant color is brown because that's the color of the heterozygous frog. They're always going to display the dominant trait because they have the dominant allele. Now, I know you know how to do a Punnett square, but there is a video on how to do Punnett squares if you need a refresher. But I do want to talk about dihybrid crosses. These are Punnett squares that involve two separate traits. And we had a little bit of trouble making the gametes for dihybrid crosses, so I wanted to review. So here is two traits, the one controlled by the alleles with G's and the one controlled by the alleles with B's. And so what you want to do is try to get every possible combination. So you go big G with the first little b, and then use that same G, but this time go to the outer B. Then this G with the first B, and this G, this inner G with the outer B. So you do both of the B's with the first G and both of the B's with the second G. Now, because this one was a rather simplistic setup, we got big G, little b for all four of our gametes. So that's, excuse me, that's all four of the sperm or egg would have the same combination because of the lack of variety in the individual genotypes. Over here, we do the same thing. We get little g with big B, little g with little b, then our second little g with the big B, and our second little g with the little b. So you get four combinations when you do dihybrid crosses. And so those combinations, the ones you come up with, are your gametes, your sperm or your egg, and they go across the top and down the side of your Punnett square. So you'll notice that we have a rather large 4x4 square instead of a 2x2 because we're dealing with two traits. Then what you do is you combine. So you match up what's on the top and what's on the side, keep the F's together, keep the T's together. So this is big F, big T, big T. Big F, big T, little t. And you would fill out the entire chart that way. Looking here at the end, I've got a little f, little t, little t. And what's really important is for you to be able to look at these combinations and tell me what traits will they have. So remember, if they have one capital letter, they have the dominant trait. So if they have a capital F, they have the dominant trait of freckles. If they have a capital T, they have the dominant trait of tongue rolling. So this individual here is dominant for freckles and dominant for tongue rolling. Whereas this individual here is recessive, no freckles, but is a tongue roller. And so it's good to be able to tell me which ones are dominant for both, like this individual, which ones are dominant for the first trait and recessive for the second trait, like this individual. So dominant, recessive. Which ones are recessive and then dominant, like this individual. And lastly, which ones are recessive for both traits. And so if you notice, I color coded these. I got 6 that were dominant for both, 2 that were dominant for the first and recessive for the second, 6 that were recessive for the first and dominant for the second, and 2 that were recessive for both traits. And these are out of 16. So you may be asked to identify the phenotype of one of these individuals or fill in one of these squares on the state test. Some dihybrid cross clues. If both parents are heterozygous for both traits, meaning big T, big B, little b, heterozygous for both traits, and both parents are like that, your outcome will always be 9 dominant dominant, 3 dominant recessive, 3, this should say recessive dominant, and 1 recessive recessive. So 9, 3, 3, 1. The other clue is that if one parent is completely dominant, then all of the offspring will be dominant for both traits, no matter what the second parent is. If one parent is completely dominant for both traits, all of the offspring will be dominant no matter what. So these 2 clues can help you out. All right, a couple other things that we need to touch on that we talked about in non-Mendelian genetics would be incomplete and codominant. Incomplete dominant is when neither allele is actually dominant. And so with incomplete dominance, we end up with a result that is blended. I always say throw it in the blender. Codominance is when they're both dominant, so they both show up. So co means both will show up. So if you take a look at this picture here, if you have both of the alleles that are big B, you're blue. If you have both of them that are Y, you are yellow. But if you have one of each, this is how it will be changed for each of these new dominants. You would be blue if it was dominant for the complete dominance, the law of dominance. But if this was a co-dominant trait, both would show up. Co means both. So you would get both blue and yellow. But if it was incomplete, we would throw it in the blender and blue and yellow would make green. So co and incomplete dominance, you will see examples of this on the state test. So example, which of the following is co-dominant, meaning both traits showed up? Black and white make gray. That's not co-dominance, that's incomplete. Black and tan make a tan dog. That's not that's complete dominance. One was completely dominant over the other. Black and a tan dog make a dog with black and tan spots. There it is, codominance. Both showed up. All right, you might have to do an example of a Punnett square with this. So sometimes we'll put the letters as superscripts or like exponents. So don't let that throw you. That just shows you that this is a codominant trait or an incomplete dominant trait. And you fill your Punnett square in just like normal. But now notice what the outcome is. So this cow would be red. This cow would be red. But a roan cow has one R allele and one W allele. So it is, since it's codominant, you see some of that red, which really looks brown, and some of that white. All right, let's take a look at another example. This is from the state test. It says a student studying feather color inheritance in certain species of chickens observed that the chickens have black homozygous, blue heterozygous, and white homozygous feathers. The student hypothesized that this feather trait color follows the incomplete dominance pattern. So incomplete dominance would mean that they would blend. So white and black would blend together. It says, what data would support this parent's hypothesis? Parent 1, black. Parent 2, white. Offspring, blue. Now this one I can get behind because if they both are in the offspring and that's a blend, the blue is probably gray, guys. That's a blend of the two colors. Then that would prove that this is incomplete dominance when you get a black and you get a white. So you get a black from one parent, a white from the other, and you end up that grayish color. That's incomplete dominance. The other ones, this one, white, parent 1 is white, parent 2 is black, and the offspring is black. That shows complete dominance. White and white making black, that shows a recessive trait. And blue and white making blue, that doesn't really prove anything. So this is the one right here that supports the student's hypothesis that black and white blend together to make that blue. And again, in nature, blue is typically gray. Like I have a blue Dane, but she's gray. So that makes sense. Black and white blend to make gray. All right. Hopefully you've got the gist of this, but again, here is an example of incomplete dominance. So we have straight hair and curly hair, and because it's incomplete, they blend together to make wavy hair. So if you take a look at these, 2 S's would be straight, 2 C's would be curly, but one of each would be a blend. That's incomplete dominance, and that is wavy. Just like this problem up here where the 2 blended together to make blue flowers. All right. Other things we learned were about blood types. So when we learned blood types, we learned that what's on the outside of the blood cell determines the type it is. So those are antigens on the outside. So the A type blood has the A antigen, the B type blood has the B antigen, AB blood has both, and O blood is like an O, it has none. And we learned that there are several genotypes because this is a multiple allele situation. So we don't just have big T, little t. Now we have 3 alleles to deal with instead of 2. So you can have the A allele, the B allele, or the O allele. 2 A alleles or an A and an O give you A type blood because A is dominant over O. 2 B alleles or a B and an O give you B type blood because B is dominant to O. A and a B together give you A type blood. This is an example of co-dominance. Both show up, both are dominant. And lastly, 2 little O's is the only way to get O because O is recessive to the other 2 alleles. So this is an example of multiple alleles, more than 2 choices, and codominance. A and B were codominant. So if you have a heterozygous A person, so IA, this A should be up here, I'm sorry, and little I, and you cross it with a O type blood, which is little I, look at the offspring. You get 2 offspring that can have A type blood and 2 offspring that can have O type blood. We also looked at sex-linked traits. These are traits found on either the X or Y chromosome. The Y-linked traits get passed from father to son, so we didn't study those. We studied what was found on an X chromosomes. And on these cases, you can see that females, because they get 2 X's, they're just basically normal. They can get 2 dominants, 2 recessive, or be heterozygous. And the heterozygous would have the dominant trait. Males, however, only get 1 X, so they only get 1 allele for the trait, and that allele determines the trait they have. So they have a higher chance of getting a recessive trait if it's found on the X chromosome because they only get 1 X chromosome. Secondly, males get that X chromosome from their mother. So any X-linked or sex-linked trait found on the X chromosome, a male would have got from his mother and his mother's family. So things like baldness, if a boy is concerned about going bald, he would look only at his mother because that's on the X chromosome, and his dad gave him a Y. So we can see here that the father gives it 1 X and a Y, whereas the mother gives 2 X's. And then in the Punnett square itself, you'll notice that we'll get 2 that are female and 2 that are males. And so when you're reading these Punnett squares, make sure that you are noticing which gender is there. And that's why we use the X and Y chromosome when we set up these Punnett squares. The XX is the female, the XY is the male. So here is an example. We have a color blindness across a male who is colorblind. So he is X little c, Y, and then a female who is homozygous dominant. She is X big C, X big C. And the question is, what is the chance that the male will pass color blindness on to his daughters? Well, he can't pass it on to his sons. So let's check out what the resulting Punnett square looks like. Notice that the 2 daughters, she's a carrier and she's a carrier, so he cannot pass color blindness on because the mom would also have to give an allele for color blindness, and mom doesn't have one to give. The two males are also normal because they get their X's from mom, and mom is homozygous normal vision. So again, with these Punnett squares, you'll want to use your X's and your Y's to signify each parent, and males are more likely to get the trait if it's recessive because they only get one X coming from mom. All right, that's all I have for you today. If you have any questions, please feel free to ask.